Hi, welcome to the lecture on environmental adaptation for animals. This is the last video in which we will discuss on the water balance. So in the remaining of this week we will focus only on the kidneys, etc. And let's first look on the different Hi, welcome to the lecture on environmental adaptation for animals. Now, this is the last video in which we will focus on the whole animal water balance. So what other organs we have than just the kidneys? And let's first look on the skin. So in the skin structure, we have different layers. You can see that this layer there on the top is completely different from this black layer. That is completely different than this with a lot of these black dots. And then this lower layer, so dermis, is completely different. So there are quite large blood vessels, and there is also some connective tissues, etc. And this outer layer, though, so this stratum corneum, can be thin or thick in different areas, and it contains mainly dead cells. So when these cells are proliferating and modifying this stratum corneum, they are becoming dead. They are releasing keratin, and they form actually lipid envelopes. So there's a cell with no nucleus anymore, and it will dead, and then you have only these lipids. And it's bonded on the cell membranes are bonded in the highly lipid environment, so we can have also lipids there around them and the keratin. And this forms a waterproof surface. So not only that, okay, we have epithelial cells, but actually these cells there's a plenty of lipid environments there around them. So there's no water that is going through. And animals in the terrestrial environments, they are divided in two groups. One is humidic and one is xeric. And the difference in humidic and xeric animals is that these humidic animals, they either lack completely this stratum corneum, or they have less developed. For example, the frogs, they have this same structure, but it's more thin, and it doesn't, so it's not so well organized. So therefore, they dehydrate easily. Okay, when we have the lipid surface, we have learned from the videos where we're discussing on the acclimatization and acclimation, or that okay, temperature is affecting. So what the compound of this lipid surface is having cholesterol, free fatty acids, and ceramides. And if we are insects, then we have hydrocarbons and just wax esters. So anyway, anything that is hydrophobic. And these, they prevent the water loss much better than any macroscopic structures. So in quite often, people think that, okay, when we have lizards and snakes, because they have scales, that scales are preventing the water loss. But that's not the case. Because between the scales, you always have holes. And if you think on the structure, on the scale of the water molecule, it's a very small. So therefore, we had to have on the molecular level, on the this hydrophobic layer that is preventing. And what we have learned from the acclimatization and acclimation elsewhere in this course is that, okay, the lipids they are changing the structure in different temperatures. That's why in animals that are tolerating well cold temperatures, they have different cell membrane in the winter time and then in the summertime. but also that, okay, if we are having very high very high temperatures, then you can see that in this, for example, in this grasshopper, so above 40 degrees of Celsius, the rate of the water loss is increasing. And it depends on different animals. So in some animals, they are starting to increasing it even in the room temperature. But in the humans, they've been testing that, okay, this phase transition is happening over 100 degrees of Celsius. And the main difference happens in plus 300 degrees of Celsius. So it doesn't have anything with the physiology. Okay, so what about then on the species-dependent variation on the resistance against evaporation? So it's quite often described and analyzed as a resistance. So you have a layer, you have water that is trying to pass through, but then some of it pass through, but majority is not passing, so it's resisting. And the resistance is related on the conductance, that is describing that, okay, how much is actually flowing through. And we have the water flow that we can measure, that, okay, how much is passing through, but it depends on the water content difference. So how much water you have in the body and how much water you have there outside. And therefore, it's a little bit more complex equation, but it's not highly difficult. And the interesting part is that the scale of the resistance is very odd, seconds per centimeters. And what we can see in the frogs and toads, the resistance is very minimal. So the water is passing through actually freely. And then in the xeric animals, they have much higher resistance, so water is not passing through. But you can see that it's very high species-dependent differences. Like in the birds, it can be 30 or it might be 200. In the some lizards, it might be 100 or 1000. And so there are easily, among different animal groups, there might be tenfold differences. In the humans, we have quite much in the average, so about 400 is the resistance. So it's 400 means that, okay, it's 100 times more resistive for this evaporation than the frog skin. Okay, how this evaporation variation is happening. So, of course, the dehydration is high in the hot and dry environments, because in the hot environments that affects on the vapor pressure, and in the dry environment you don't have so much water in the environment, so it's easily going out, and therefore there is adaptation. So the southern populations in the grasshoppers, for example, have less evaporating cuticles. So this might be that this is on the evolution and adaptation. So slow process, something related on the gene activities and gene structures, and then affecting that, okay, how much lipids you have there on the surface. But then, in some cases at least, they've been showing that there's also acclimatization. So the same animals might have also seasonal differences, for example. So in the summertime in the desert, it's a high temperature and very low humidity, but then in the wintertime it might be a little different. Okay, what about then, not only on the skin, but we were discussing earlier that, okay, when we are having the breathing, that is also a problem for losing the water especially in the terrestrial environments. So these xeric animals, we have one thing common. So we have the respiratory system is inside our body. So we have the lungs there inside, of course, but it's important thing is that, okay, now we can regulate. If we would have the all these bubbles there in where we have the respiration, if that would be just on the open field, the evaporation would be so fast, so there is no savior anymore. And when we have warm blood, the inhalated air is cold and the exhalated air is hot. And why we have the exhalated air hot? First of all, in the lungs, the air is saturated with water. So there is plenty, as much water that can be in this temperature according to water pressure. And in cold temperature that we are in-breathing, there is always less if the temperature of the environment is colder. We were in previous videos we were discussing okay, what happens on the vapor pressure in different temperatures. And in several mammals and birds, they can cool down the exhalated air in pre-cooled nasal cavity. This is not very common knowledge in humans because we are not doing this. But the idea is that, okay, if we think on the respiratory cavities, it has the epithelial or whatever structures there, and when you are having cool air is coming, it will cool down the nasal cavity. So these epithelial cells, etc. And therefore, when the exhalated air is passing through, it will be cooled because we have the pre-cooled nasal cavity that is cooling down it. And in some cases, this air is warmed only by few degrees during the respiration. So over here we have the ambient temperature, and this is the temperature of exhalated air. In the dogs, it's way below 37. degrees of Celsius, that would be otherwise. So for example, in room temperature, the air temperature is about 25 in the dogs. But in some animals, it's only 20. So they are cooling down to say. So that means that, okay, now might be that if there's enough water in the environment, in the air, so it's very humid, then you are not losing at all. And in the typical case, we don't have very high humidity in the environment, but anyway, you are reducing the evaporation because you are cooling down the air. Okay, what about then on different animals? So over here is the weight specific evaporation of water loss. So on the procents to compare the body weight per day. So we have hummingbirds that are losing over 30% of their body weight in a day. In just that, okay, they are losing water. And in the other end, we have animals that are not losing much at all. And what we can see that, okay, in these small animals, they have high evaporation rate. And partly it might be related that, okay, they have high metabolism. But on the other hand, the high metabolism means that, okay, you have producing from the glucose to ATP and carbon dioxide and water. So you are also producing metabolic water, but that's not compensating it. And what is the large difference between these two birds is that the small birds, they have much more surface to compare the volume. And that is the key for why these small animals, they are losing a lot of water to compare the large animals. Okay, let's look still one more time on the different kind of animals, animal groups. So over here we have mammals and birds. The typical evaporation of water loss might be 0.5% of the body weight within an hour. And it's much larger than in the ectothermic lizards, because these ectothermic lizards are not so much related on the oxidative phosphorylation. They are not they are not needing so much oxygen for the from the respiration, so that's why the evaporation is lower in the lungs. And it's still much larger in these humidic vertebrates because they are losing the water through the skin. And this can be quite important thing, that now, for example, these frogs, if you are losing 1.5% of your body weight within an hour, that means that, okay, this means that it's over 30% of the body weight per day that they are losing. So that's why these frogs and toads, you usually see them when there is water around. Okay, and let's compare to completely different group, like these semi-terrestrial crabs that are sometimes on the terrestrial environment, sometimes in the aquatic, and they typically have maybe 1%. So it's quite much on the same scale with the warm-blooded vertebrates. And the main difference is that, okay, these crabs, they might have that the skin resistance low, but at the same time low oxygen need. So the respiration is lower also, and that is the problem in our in these mammals and birds, that because we have high body temperature, we need so much oxygen. That's why we have much higher evaporation of water. And then in different invertebrates, we have few insects that are very good in saving water, but this is not very typical. And so they are losing maybe 0.5%, but in the other end, we have these isopods like the pillbug over here, that they might be losing several percent of the body weight. And if you are losing, for example, in this 10% of your body weight within 1 hour, okay, if we multiply this with 24, that means that, okay, you are losing over 2 times your body weight within 1 day of water. And therefore, we might have seen quite often these isopods, but these are underground. Because they are losing so much water. Okay, what about then on the kidneys? Because this urine is always the typical case that we think that, okay, that is the way how we regulate the amount of water loss. And we use, in humans, we can have very wide range on the urine osmotic pressure. It can be 0.1 or it can be 4. So it can be 4 times higher than in the plasma, or it might be only 10% having other things than water to compare the plasma also. And this is not the typical case. So it happens in mammals and birds and insects. And in the insects, they have quite typical urine osmotic ration about 2 or 4. So they can have 2 times more solutes or 4 times more solutes than in the plasma. And in the mammals that are living in the dry areas, they can have very concentrated urea. So in the humans, when we are not drinking enough and we want to get rid of as much waste products, etc., then we might have that, okay, the urine might be a little bit over 1,000 milliosm per kilogram. So it's 4 times more than in our plasma. But then in these hot environments, we have the camels that might have 8. Then we have kangaroo rat, and then Australian hopping mice. That's the extreme. So now this urine is very concentrated with different substance than the water. And let's look a little bit on that. Okay, if it's 1,000 milliosm, so that means that, okay, it's 1 to 10 osm. And what we were discussing earlier, that, okay, well, how this related, it's not directly, but we can calculate that the osmotic pressure by just getting the sum of different ions, for example. And if the concentration, if the osmotic pressure is 10 osm, that means that, okay, we could have sodium and chloride 5 plus 5 mole per liter. And that would then mean that, okay, then we have 5 mole per liter sodium chloride. And this solution, you can do it. It's still diluted, but in this, whenever you have anything in more than 1 mole per liter, the solubility is becoming the problem. So this is actually having as much salt as you can dilute. So it's saturated. So that's why it cannot be much higher anymore. In the birds, it's still much lower. So it's in typical, it's only about the same, as much solutes as in the plasma, or 2 times more, or something like that. So they must have some other mechanisms to save water. And these other mechanisms can be that, okay, you have different kind of nitrogen waste product. We will discuss these on the next videos. Or then with the dormancy. So when you are dropping your body temperature, that of course means that, okay, then you are reducing the metabolism, then you are reducing the respiration, and therefore you are not losing so much water. And of course then you can also be in some kind of shelter. The other way is not just to look on the evaporation itself, but in some cases there can be studies in which they describe water turnover. This turnover is, you are understanding, studying how much water is gaining in the body and how much is losing. And this is on the daily activity. That means that, okay, these animals can just move free, do whatever they want, and then after 24 hours you will record it again. So the idea is that, okay, you are injecting a labeled water. Usually this means that, okay, the labeled water you have you have one hydrogen is a little bit radioactive, so then you can measure it. And you can catch the animal after 24 hours and check that, okay, how much this labeled water still exists. And it's higher in the small animals because they have larger metabolism, they have also larger surface area. But what we can see that it's in completely different scale in different animal groups. So in the mammals versus the reptiles, this difference is about tenfold. So meaning that if these reptiles are losing perhaps 1 or 2 milliliters per day, we mammals, we are losing 40 milliliters per day. And these birds and marsupials, there's about 3 times higher. So they need maybe 30 milliliters per day in the same size of animals. And then you can see that, okay, the large birds or large marsupials, they are becoming a little bit more effective than to compare the other mammals, but never as low as these ectothermic reptiles. Thank you.